Good morning, this is Kavita Kundra and we are here again with Mscdfsm Biochemistry. The chapter today is hormones. The endocrine system is composed of endocrine gland whose secretion pass directly into the bloodstream. The secretion is called hormones. Hormones are the substances that is synthesized in one organ and transported to circulatory system to act on another tissue. Since hormone carries signals or messages, they are also called messengers or specifically first messengers. Regulation of endocrine system. Regulation originate in blood and central nervous system. It receives inputs from many internal and external sensations like hunger, danger, dietary intake, etc. It is also called hormonal cascade system that shows chain of command in hormonal signaling hierarchy. The signal first originate in external environment and transmitted as electric impulse to hypothalamus, and the hypothalamus release hormone in nanogram. The releasing hormone is carried by blood to anterior pituitary, and this is the first target. Anterior pituitary secrete tropic hormone or tropin in microgram. Then this is then carried out to endocrine gland to get stimulated. Now the endocrine gland synthesizes its specific hormone in milligram, and then this hormone travels to the target tissue through the blood. So the starting point is hypothalamus, the ending is danger target tissue, and hypothalamus is coordinator. It receives the integrate messages from the central nervous system. Infections in the body. Suppose a person is suffering from the infection or hemorrhage. The person perceives fear or pain. Electrical chemical signals are generated that are received by CNS. CNS pass signal to hypothalamus. It releases corticotropin releasing hormone that goes to anterior pituitary. It in turn secrete adrenocorticotropic hormone that goes to adrenal cortex and triggers cortisol. The cortisol alter metabolism in liver cells and hepatocytes. Cortisol increases the rate of gluconeogenesis. This ensures that the body gets enough fuel to overcome the adverse action. It is clear that at hormonal signaling hierarchy at each level, a small signal elicits a large response. The electrical signal releases CRH that in turn release corticotropin. Corticotropin act on the adrenal cortex to release cortisol. It is called hormonal cascade system. Cascade mechanism means that small amount of extracellular compound can affect large number of intracellular enzyme without crossing the plasma membrane or binding to each target protein. Whenever there is increased level of products at any stage, the feedback inhibition is initiated, and this ensures that the product is made only until the necessary concentration is reached. Mechanism of hormonal action. There is two types. First one is the target cell concept. There are 200 types of differentiated cell in the human. Only few hormones. All cells are the targets of one or more than 50 hormones. Hormone receptor. Hormones are present at very low concentration in extracellular fluid, and these concentration is lower than the sterols, amino acid and peptide. Hence, the target cell must distinguish not only between the different hormones but also between different kinds of chemicals. This high degree of discrimination is provided by cell-associated recognition molecules called receptors. Target cell has ability to selectively bind with a given hormone to specific receptor, and hormone bind to specific receptors. Hormone-induced action are terminated when the hormone dissociates from receptor. Characteristics of the receptor. All receptors have two functional domains: recognition domain that binds to the hormone and a region that generates a signal that couples hormone recognitions to intracellular function. The coupling occurs in two ways depending upon the chemical nature of the hormone. Polypeptide and protein hormone and catecholamine binds to the receptor located in plasma membrane and then generate a signal that regulates intracellular functions that changes the enzyme activity. Steroid, retinoid, and thyroid hormone react with intracellular receptors and hormone receptors complex directly provide the signal to genes whose transcription rate is affected. These hormone receptors have functional domain that binds the hormone, that binds to specific DNA region that is involved in interaction with other co-regulator protein, results in activation of gene transcription, that bind to one or more protein that influence intracellular trafficking of the receptors. Both recognition and the coupling domain occurs on the receptor. Classification of hormones. There are different types of hormones according to the mechanism of group 1 hormone that binds to intracellular receptors: thyroid hormone, glucocorticoid, mineralocorticoid, retinoic acid, calcitriol, androgens, estrogens. Group 2 hormones that bind to the cell surface receptors. There are different categories. A category, the second messenger is CAMP, glucagon. Calcitonin, parathyroid hormone. A2 adrenergic catecholamine, androgenic catecholamines, thyroid stimulating hormones. A2 adrenergic catecholamines, adrenergic catecholamines, thyroid stimulating hormones, anti-diuretic hormones, adrenocorticotropic hormones, human chorionic gonado. B part, the second messenger is CGMP, atrial natriuretic factor. The second messenger is calcium or phosphatidyl inositol or both. Acetylcholine, A1 adrenergic catecholamine, oxytocin, antidiuretic hormones, cholecystokinin. Second messenger is kinase and phosphatase cascade, insulin, growth hormone, prolactin. Group 1 hormone, they are lipophilic, they are fat soluble, and these hormones associate with plasma transport and carrier proteins. These overcomes the problem of insolubility in aqueous medium of plasma. At the same time, it prolongs the plasma half-life of hormones since they cannot be eliminated easily when bound to protein. Binding capacity and binding affinity determine the relative percentage of bind and free hormone. The free hormone in the active form crosses the lipophilic plasma membrane of the cell and the receptor for the group 1 hormone present in cytosol or nucleus of the target cell. Then hormone receptor complex is formed. This complex, the ligand receptor complex, is formed, and it is intracellular messenger that conveys the message brought from CNS to specific cell. Group 2, they are hydrophilic or water soluble. They cannot cross the hydrophilic plasma membrane. They bind to plasma membrane cell, and their receptors are located in the plasma membrane. Such hormones that binds to the surface of the cell communicate with intracellular metabolic process through intermediary molecule called second messenger. The hormone is called first messenger and second messenger are generated as consequences of ligand receptor interactions. When epinephrine binds the plasma membrane of the certain cell, the concentration of cyclic AMP increases. The characteristics, and due to that characteristics, and based on that, we will be differentiating in group 1 and group 2. Characteristics is type of hormone. So in group 1 we have steroids, retinoids. In group 2 we have polypeptide and protein. Solubility. In group 1 we have lipophilic and in group 2 we have hydrophilic. Transport protein. In group 1 we have group 1 hormone can transport protein, yes, and group 2 no. Plasma half-life, long and short. Receptors, group 1 has intracellular and group 2 has plasma membrane. Mediator, receptor hormone complex, and second messenger CAMP. Thank you so much, and please go through the chapter. It's easy, not at all difficult, and please share it with your friends, and you can help more and more students to study MSc DFSM during this corona time. Stay safe, stay protected, get vaccinated. Thank you so much. Bye bye.